As your previous videos have suggested, eukaryotic mRNAs are processed or modified prior to leaving the nucleus and entering the cytoplasm for translation. I've separated that RNA processing into two separate videos, this one describing the first and the last of those modifications. As the Khan Academy video showed you, there is a difference between the transcription process in prokaryotic cells relative to that of eukaryotic cells. In the image on the right side, you can see that an RNA transcript that is being produced in a bacterium can begin to be translated even before the transcription process is complete. That is because there is no spatial separation between where the DNA is located and where the ribosomes are located in a prokaryotic cell. That's not true in a eukaryotic cell, the image on the left, which demonstrates that transcription and RNA processing are going to occur in the nucleus, and it's only after RNA processing is complete that the mature mRNA can be translocated from the nucleus to the cytoplasm, where translation then will occur. That RNA processing is a three-step process, and as I said, we're going to talk about the first and the last of those three steps in this video. The first and the last steps are RNA capping and polyadenylation. The RNA cap occurs at the 5' end, and polyadenylation occurs at the 3' end in the form of what is labeled here as a poly-A tail. Both 5' capping and 3' polyadenylation serve the same functions. Both contribute to stabilizing the mRNA, extending its lifespan once it leaves the nucleus. Both also are helpful in the transport of a mature mRNA from the nucleus to the cytoplasm. And in addition to that, both the 5' cap and the 3' poly-A tail facilitate or make the process of translation much more efficient. We'll start by talking about the 5' modification, RNA capping. As the Khan Academy video described, the 5' cap is a modified guanosine or a modified G ribonucleotide. The modification on that guanosine ribonucleotide is the addition of a methyl group in the number 7 position of the nitrogen-containing base. This is referred to as 7-methylguanosine, or shortened into 7-methyl-G. This 7-methyl-G nucleotide is attached to the 5' end in an upside-down sort of way, with the 5' end of the 7-methyl-G attached to the 5' end of the mRNA transcript. The 5' ends of these two ribonucleotides share what is called a 5' to 5' triphosphate bridge. While addition of the 5' methyl G cap can occur as soon as the enzymes responsible for that modification are able to access or see the 5' end of an mRNA, processing at the 3' end cannot occur until the transcription process is complete, until the 3' end of the transcript has been produced. While we focus upon the polyadenylation, prior to polyadenylation, a cleavage step has to occur. Cleavage will occur approximately 10 to 30 nucleotides following a specific AAU AAA sequence in the mRNA. The following diagram depicts all of the steps that are required in order for this cleavage and polyadenylation to occur. Here, the RNA polymerase is shown as the blue blob, and it's reading a DNA template strand and producing a blue mRNA. On the tail, if you will, of the RNA polymerase, you can see two enzymes indicated by green and brown ovals. Those enzymes are cleavage and polyadenylation stimulating factor and cleavage stimulating factor. As soon as the AAA has been transcribed, the RNA polymerase keeps going, but these two enzymes are transferred from the tail of the RNA polymerase to the mRNA. As I said, 10 to 30 nucleotides after that AAA sequence. These enzymes cleave the mRNA, and another enzyme, poly-A polymerase, will hop onto the cleaved end of that mRNA and begin adding adenosine ribonucleotides to that cleaved 3' end, thereby creating the poly-A tail. Several hundred adenosine nucleotides can be added, and in fact often are added, to the 3' end of an mRNA. Note that part of the stabilization that this modification confers is dependent upon poly-A binding proteins, these green ovals, that bind to the poly-A tail. These proteins, poly-A binding proteins, binding to the tail, prevent access of enzymes that would destroy the mRNA. While the previous image showed you the cleavage and polyadenylation factors being bound to the tail of the RNA polymerase, those aren't the only enzymes that are bound to the tail. This diagram shows you that the capping factors are also attached to that tail. However, because the 5' end that is capped is modified well before the 3' end, that capping factor has hopped off the tail and onto the 5' end of the mRNA as soon as that 5' end is accessible, and the capping factors transfer that modified guanosine nucleotide to the 5' end of the mRNA. To summarize, both the 5' and the 3' modifications perform the same functions, and those are: they enhance the stability of the mRNA, they allow for the mRNA to be exported from the nucleus to the cytoplasm, and they make the process of translation much more efficient once that mRNA is in the cytoplasm.